Hi, I'm Cindy Chestek. I'm an associate professor of biomedical engineering at the University of Michigan in Ann Arbor. Today I'm going to talk about carbon fiber electrode arrays. I am going to start by going through my favorite application of neural probes because I can't resist talking about motor neuroprosthetics. So I'm going to give that as an example of usefulness of having a very high density array of neural probes. Then I'm going to talk about the history of carbon fiber array development and some of the literature that led up to some of the work that we've been able to do. And then finally, I'm going to talk about the carbon fiber electrode arrays that we have developed as part of our Brain Initiative work as a neuroscience tool. So let me get started by talking about motor neuroprosthetics. So what do I mean by that? Specifically, I mean that we take signals from the brain, the nerves, or muscles, and we want to use those signals to try to extract a motor control signal. So for example, in the case of amputation, we might want to take a signal from the nerves or muscles and use that to control a prosthetic hand. You could also imagine the case of spinal cord injury, where we want to be able to get signals from the brain and use that to control electrical stimulation of a paralyzed arm. So let me go through an example of each of these and talk about what electrodes are used today and where there's opportunities to do better in the future. So let me start with nerve-controlled prosthetic hands. So The Empire Strikes Back came out in 1980 and had this really compelling science fiction picture of nerves controlling a prosthesis. In real life, it's unfortunately been really hard to get a viable control signal directly from a nerve. And the reason for that is, despite lots of very intelligent, hardworking people trying to get that signal. It's just that the physics is not in your favor for doing that because the nerve is just so small. You're talking about nerves that are about 1 millimeter in diameter, and you're going to have to stick a wire inside of that nerve in order to pick up that signal. And so, this can be done in fairly controlled or at least short-term conditions. But, even then, the signal is really small and it's hard to get a realistic signal in a person walking around. And you're very limited by how many electrodes you can currently put into a nerve because it causes a scarring response, which I'll talk more about later. In my group, I think we decided that it was so hard to get a signal from a nerve, we sort of found a way around it in the case of amputation, where because we can't record from that nerve, we instead attach a small piece of muscle to the end of an amputated nerve. And you can get multiple signals this way by dividing the nerve down into different branches. But what you really gain from wrapping a piece of muscle around the nerve is that the nerve regenerates into the muscle and creates a much bigger signal inside the muscle. But you also gain on the electrode side because now I can use an electrode very similar to what a cardiac pacemaker uses. So now that I have a centimeter of muscle, I can shove a 1 millimeter probe inside of it. But this way it's limited because I can only get one signal for each nerve branch. But it's actually pretty good in terms of being better than what's state of the art. So I'm going to go ahead and play a video of what we can do with just 2 signals recorded from nerves. And if you think about it, you can only move your thumb based on muscles that are inside of your hand. And so this is something we have to get from nerves.
Go. Stop. Go. Stop. Go. Stop. Go. Stop. Go ahead. Stop. Go down. Up. Over. Go down. Up. Over.
Okay. so that's an example of what we can do if we can get two independent signals out of the nerves. Imagine what we could do if we had even more signals, because we had smaller electrodes and we could put more of them inside the nerves and muscles. So let me talk about another application where we're using electrodes to try to get prosthetic control signals. And so not only can we do this from the nerves and muscles, we can also go directly to motor cortex. And in the case of spinal cord injury, this is where you have to go to get good control signals to control the arm and hands. So what you're seeing there is, it looks very dramatic, but in real life it's about 4 by 4 millimeters, and it goes about 1 millimeter into the brain. This is called the Utah array, and it does a pretty good job of recording neural signals, and can last for multiple years. Let me show you a picture of what the surgery looks like there. And I would say this is a pretty darn good electrode. I'm going to tell you about new electrodes, but this does a pretty good job of recording signals from the brain, and it absolutely generates a scar. But every video you've ever seen of somebody controlling something with a Utah array has been done after the period of scar formation. It's been in, according to a recent review article, it's been in 48 human brains. So it has a really good safety record. In my lab, we use signals from Utah arrays to control finger movements. So let me play a video here of a monkey who has one of these Utah arrays in primary motor cortex. And he's going to use those signals to control an animated index finger. So the moving finger is being controlled directly by brain signals. So he also knows how to do this task with his real hand. Okay, so I mentioned before that those signals were for multiple months after implants, which means that they've already generated what every conventional electrode does when you put it in the brain, which is it generates a scar. So to sort of motivate why we need new probes, let me talk about what that means. So the Utah array has elements that are about 80 microns across. And for electrodes, are around that order of magnitude, made by multiple vendors, if you take that and put it into the brain, you are going to generate a scarring response. The brain protects itself from foreign bodies, and it detects that as a foreign body. And so what does that mean to us if we are trying to record neural signals? So on the right is a figure from the Tresco lab, and they're looking at the health of the neurons and neurofilament after a long-term implant. So right in the middle of the slide, if you can see the orange bar, that's the location that an electrode was in. And then in the top right plot, you see lots of blue dots. And those blue dots correspond to neurons. And unfortunately, you can see, on the right, there's a lot of blue dots. But as you get closer and closer to that orange bar, there's many, many fewer blue dots. And probably something very similar happens when you put a Utah array in the brain. Again, it still has, the best safety record in human beings. It's been implanted many times, but this is the reason why we would not put this in a healthy brain. Okay, so that's a major problem with many different kinds of electrodes. And in fact, I have to go 400 microns away to get away from that zone of scarring. And indeed, the Utah array has a pitch between the different electrodes of 400 microns. So if I want to get more signals, so I want to control more of the fingers or generate, control multiple parts of the body, I can't just make my Utah array more dense. If I start putting those closer together, I'm eventually going to turn the brain into a solid block of scar tissue. So that's not a good path forward. Okay, so before I talk about how we're going to try to solve this problem, let me just talk a little bit about specifically why is scarring a problem. I think there's sort of a misconception that the process of scar formation insulates the electrode, like that it electrically insulates the electrode. And I think, that does happen, but that is not the important thing that happens. So specifically, let's look at how these signals get to our electrode. So if I have the black dot there is an electrode, and I am recording from a field of neurons, I can think of those neurons as lots of current sources. As ions get sourced and synced into different compartments of the neuron, I'm going to pick up an external voltage that corresponds to those current sources and sinks. And it's actually, it's a fairly simple relationship. So, it depends on the current, so that's I naught. It depends on the conductivity, which is that sigma. And it also depends on the distance to the electrode, which is that R. And so, with a scar, you're definitely going to increase the resistivity or decrease the conductivity. But first of all, that it's not that big a deal. It's still tissue, so it's not exactly as insulating as rubber. And we're actually talking about a pretty high impedance probe to start. So the impedance associated with the scar, isn't necessarily a big problem. We can handle the impedance of the electrode. The big problem that we have, however, is the distance. So as a scar forms, it's going to push those electrodes away. And as you see from the equation, the voltage is inversely proportional to the distance R. And 1 over R means that signal falls off extremely quickly as we move away from the electrode. So that movie shows how far this signal extends from the middle of the neuron. So the white dot is a neuron, and the field that you can actually record doesn't extend for more than about 25 microns on each side. And this is from Holt and Cook, 1999. So this is a real problem if I'm trying to record from neurons. If that scar, even a small scar, can push me 20 microns away, which is going to make my signals much smaller than they would be otherwise, and a large scar, say 50 to 100 microns, is going to push all of the neurons outside of range of my electrode. So I have to somehow deal with that scarring response if I want to put my electrodes closer together, and ultimately to put more electrodes into the brain. And I should say what this means is that all previous electrodes have, in a sense, been operating with one hand tied behind their back, because the scarring sets in, and it's it's known that this happens, and it generally happens before most experiments are done on the electrodes. All right, so that was the state of affairs around 2005, right, where we didn't have any idea how to deal with the scarring problem. And, it was kind of a dark time, because it was really unclear, if there was ever going to be a way to stop that foreign body response, or at least minimize that foreign body response, so that we can record from these neurons for a long time period. Okay. So I think a solution to this started emerging in the literature around 2007. And there were two very similar studies, one published by John Seymour in the Kipke lab, and the other study published by Skozin et al. In Pat Tresco's lab. But the results are very similar and support each other as being a possible solution. So what you're looking at is two conventional silicon probes, except that they have a lattice element. And what's important about that lattice element is that the individual components of that lattice are 15 microns or smaller. And at that size, it's actually smaller than a neuron. And what they found is that the number of neurons surrounding the electrode was much, much higher around these small elements. So for example, taking this from Skozin et al., around the 15 micron lattice elements, there was 85% of normal neuron density, compared to the solid part of the electrode, where there was only 35% normal neuron density, which was pretty typical for a probe. So this is an enormous effect. There are 50% more neurons of the neurons that we're missing around these small elements. And this really suggests that, what we should be doing is making these electrodes very, very small. So that's great. But if you'll notice that if I took those lattice probes and if I stuck 100 of those into the brain, they're actually doing damage on the way down. So they're still sort of have the profile of a regular electrode. So that's, ripping up circuitry that I would rather not rip up. And so the key is going to be how do we put something 15 microns or smaller into the brain, and do that in a way that that's the only thing entering the brain and it does very little damage on the way going in. All right, so my lab and others is attempting to solve this problem using one of the strongest materials that we have access to as engineers. And I specifically mean carbon fiber. I took this from a textbook, and it does a really good job describing the cool materials properties we get from using carbon fiber. It has high strength tensile stress, high Young's modulus, it has good fatigue resistance. It's actually really electrically conductive, so it serves really well as a wire. It also has good chemical stability, and excellent resistance to acids, alkalis, and many solvents. And I won't get into that here, but that actually is a really important process, a really important thing for surviving in the brain, maybe one day for decades. So short version of that, it's strong and cheap, certainly at low volumes. And the other thing is that it's a really, it's an easy material to handle. And despite being really hard, like being very stiff, and this is going to help us insert into the brain, it's also very durable and harder to break than most of the other common electrode materials. So I want to play a video here of carbon fibers embedded in silicone, and we're going to run them over with a glass capillary, which we can do 10,000 times without breaking the electrodes. Okay, so admittedly, that's from when we embed it in silicone. If we put it on something stiff, it's going to be a little bit more breakable. But in general, this is a material that is much easier to work with and it will survive surgical handling pretty well. So first of all, I should say that carbon fibers have been around for neuroscience for a really long time. Particularly due to the seminal work of Mark Whiteman, they're commonly used for recording neurotransmitters, and this can be done with something called fast scan cyclic voltammetry. So, these were around a lot of neuroscience labs, but the way that they were used is, these are closer to the size of cobwebs than hair, so they're very small. And so they would normally be put inside of a glass capillary, and that makes them easy to handle and insert into the brain. The first singulated probe, by which I mean, just the carbon fiber. And here we're talking about starting with a 7-micron carbon fiber. The first one was developed by T.K. Kozai, and we still use a lot of his manufacturing processes in the lab. So how you build these is you start with a bare carbon fiber. And in proportion, neurons are universally bigger than 7 microns. They can vary in size, but like 10 to 30 microns is the size of the neuron, and our carbon fiber is going to be much smaller than that. So it gets coated with a layer of Caroline C, and that's used as an insulator because I don't want to record from the whole carbon fiber. I just want to record from the tip. And then the tip is cut, and we now do that with a wide variety of techniques. And in TK's initial paper, this was done by PDOT. There was also a layer of PDOT added to reduce the overall impedance, and that makes this a much better electrode. And that's what it looked like in his 2012 Nature Materials paper. So this is a very, very small electrode. And you could kind of get it into the brain with tweezers, with a lot of coaxing. And again, you can do that because it's carbon fiber, even though it's so small. But it was fairly challenging to try to put multiple of these electrodes in any given surgery. So in my lab, we developed this into an array. So specifically, what you're looking at on the upper left is our sort of model device from 2015 that we use to demonstrate a workable geometry. And I should say this was developed by my former PhD student, now postdoc, Paras Patel. And this followed several years of having cobweb-sized electrodes flopping uselessly against the surface of the brain. But, after a lot of trial and error, we sort of figured out that, 500 microns is the magic number, where if you can take a 8-micron carbon fiber and you make it 500 microns or shorter, at that point, you get 100% success in penetrating the pia of the brain, which is the top layer of a rat brain. So then we came up with a number of ways to temporarily or permanently have 500 microns of carbon fiber. And so what you're seeing there is 500 microns of carbon fiber at the tip, and then there's gradually widening silicon, just so we can get, the other 500 or to 1,000 microns into motor cortex. And I'm going to play a video here where you're just going to see the carbon fiber disappear past the surface of the brain. And at least for that first part of the video, the brain barely reacts. All right, so that was one way of making the carbon fibers 500 microns or less. Another way that was really useful in a lot of applications is we put a dissolvable material on the electrodes. So you can see that pictured on the left. And then, you can penetrate the brain with the first part, and then you can melt off the rest of the material as you go. And so I should say that basically the important thing is to penetrate that top layer called the pia. Once you're through the pia, you basically have your pilot hole, and it's holding the carbon fiber in place. And you can actually drive a carbon fiber all the way through a brain at that point. So it's just that first layer of penetration that's really important. And I should also say that longer than 500 microns, carbon fibers do penetrate, but it's not 100%. And if you want to put an array of closely spaced electrodes, you need it to be absolutely 100% of the electrodes that penetrate, because if one of them goes sideways, it can take out the rest of your array. Another device that we do a lot of work with is pictured on the right. That's kind of our workhorse device, where we just have carbon fibers projecting off the end of the circuit board. And we can actually put that in a cannula. So that lets us get anywhere in the rodent brain, where the cannula is obviously going to do some damage. But, the carbon fiber extends 500 microns past that. And so, typically that's enough to get outside of the damage radius of the cannula. And a lot of the, the early results we've seen or in recording from neurons came from these workhorse devices. All right, so let me talk about some of the capabilities of these probes. So they're useful for recording action potentials, but there's other things that they can do that would be very difficult to do with a conventionally sized probe. And all of these beautiful images I'm going to show you were done in Dawen Tsai's lab. So what you're seeing at the top there is a rat brain where we have done the slicing with the electrodes in place. So remember, these are smaller than neurons. And so while it's really hard to slice a normal size probe, because you're either going to break your blade or it's going to move the tissue as you break it, it's really hard to leave that in there. And people usually pull the electrode out, and then they find it based on the scar. For carbon fibers, we can just leave them in. And as we slice, they appear as little black dots. What you're looking at there is a light field image from the microscope with a lot of black dots. And I should say that, that the little flecks of carbon, sometimes they stay in the tissue slice, sometimes they don't. The key is actually knowing where they are. Because once you know where the hole is, you know which hole comes from a carbon fiber rather than a capillary, it's really obvious to follow. So as soon as you know the location or roughly the area that it should be, you can find the hole, and my students can circle the hole all the way down to the tip of the electrode, and it's just slightly larger than the carbon fiber itself. So now we can take those black dots, and we know where the electrodes are on our other images. And now you can combine these slice-in-place electrode positions with any staining of your choice. And as part of our Brain Initiative project, we're trying to put together a menu of stains that you can use in combination with the carbon fibers. So red dots here are electrodes, and the green dots here are neurons. And importantly, this is 11 weeks after implant. So any scarring process that was going to happen should have set in by now. So you can see here's two electrodes. They moved a little bit in implantation, and they ended up about 30 microns apart. And there's two neurons settled in between them at 11 weeks after implant. Again, I mentioned we can circle these holes all the way down, and we believe we can identify the tips of the electrodes because these holes disappear pretty quickly. And we've used that to start doing 3D reconstructions of the neurons surrounding the electrode tips. So let me go ahead and play a movie of what that looks like. Okay, so you can see that those neurons were pretty close and personal with the electrode. We can also look at this across, hang on. We can also look at this across 18 electrodes with an average of 11 weeks after implants. And we can look at the neuron density as in different rings surrounding the electrodes. And you can see here at 0 to 25 microns from the electrodes, we have a really nice density of cells. There might be some, small decrease right near the electrode, but so far in 18 electrodes, it's not statistically significant compared to control brain, which you can see at 100 microns plus away from the electrode. And I think that, while we're really focused on neuroscience applications of this probe, this does strongly suggest that there's going to be safety implications if we can eventually use these electrodes for a variety of clinical applications. There's just many fewer missing neurons within 150 microns of the electrode. Another point, let me just go back a little bit, I want to make for neuroscience, is that traditionally the world of recording from neurons and taking beautiful images of neurons ex vivo have been pretty separate. So, you can map out what neuron is connected to what neuron using these really cool emerging microscopy techniques. You can also record in the lab from a freely behaving rat and figure out the relationship of, those spikes to behavior. But so far these worlds haven't come together. So we're really hoping that we're going to be able to look at these images and those 3D positions of the neurons. And while we know it's going to be difficult, we're really hoping that we can associate particular waveforms with particular neurons and eventually be able to record from a neuron and then see what other neurons in the brain it connects to. I should also say that the yield of neurons that we're getting, you'd expect the yield of the neurons to be very high if you're having these very high densities near the electrode tip. In some of our earlier histology work and some from other groups, there was very, minimal damage to the neurons. And, we saw this in the literature before we were able to slice the electrodes and visualize them. We saw that there was very little immune response. But we always had suspiciously low yield of neurons, even though we were having such a good immune response. So my student, Alyssa Welly, figured out that this was probably because of our manufacturing of the carbon fibers. And you can see in the upper left, we switched from scissor cut to laser cut. And this was a night and day in terms of how many of our electrodes were picking up neurons. So now everything kind of makes sense where, you see a lot of electrodes with neurons very close by. And now we generally record a neuron from most of our channels. So I'm showing you acute data, and you can see our new chronic data in our upcoming paper. But looking at, 97% of the electrodes recorded a spike that was greater than 100 microvolts, and a full 24% of our electrodes recorded a spike that was 500 microvolts or more. So again, the existing arrays that we've been using are pretty good. We still use the Utah array for all brain-machine interface experiments in my lab. But I think it is fair to say that we would be recording a lot more neurons if we didn't have some of the scarring problems that were associated with conventional probes. And we really want to see this kind of yield on a, one day a human device. So we're very grateful to the Brain Initiative for funding us to do, essentially work out all of our bugs and create a range of devices that are going to be useful to people studying brain areas all over the brain. So we finally settled on doing a 3 millimeter, a 6 millimeter, and a 9 millimeter device. So there's carbon fiber at the very tip. You can probably see that there's graded, these shanks are gradually expanding silicon as we go up, and that silicon is there just to get the, enough support, for example, to get, more than 500 microns into the brain. And that 3 millimeter device is really intended for recording from cortex. where it's just good to have some strain relief on the probes with that extra sort of millimeter or so. I can tell you most of the time went into the clean room portion. So it just takes a long time to iterate on these designs where, you know, we finally sort of settled on something. So we were trying to keep all the silicon as small as possible. We think we finally settled on the design that reliably inserts every time. So where this project is at is we just made 130 devices, and we're very grateful to our beta testers who have agreed to test them, and we're implanting them in primary motor cortex. And we can make these on a variety of connectors, as we've had to for some of our collaborators. And I should say that we can use all of our techniques to make a dopamine sensing carbon fiber as well. It's a little bit different in how you prepare the tip of the electrode, but you know, this is the same carbon fiber that's used to sense dopamine. And we also went ahead and did a head stage that's appropriate to recording dopamine using fast scan cyclic voltammetry. We've also been really fortunate to be able to distribute mostly our existing workhorse devices. And this was done through the NeuroNext MINT program at the University of Michigan, run by Yusik Yoon, where I'm able to have a full-time technician who has just been making devices for neuroscience labs all over the country. So, you know, if you Google NeuroNext MINT or, you know, NeuroNext Michigan, it'll bring up the website. And you can click on carbon microthreads, and if you fill out the form, it sends a request directly to my technician. And we have a data sheet up there for devices that we have available. But I'll mention that most people who have contacted us have wanted something slightly different. My awesome technician, Juliana, has done a really good job trying to accommodate as many requests as possible. So we've so far distributed on the order of 300 devices. You can see the, you know, all of the different universities where we've sent devices so far. It's about equally, I don't know, a little bit more, about 50-50 split between our internal collaborators, which use many more devices, and some of our external collaborators who have, you know, gotten a subset of probes to try them out. But about 300 devices, and you know, we still have a few years left on this distribution program. And I should say we're hoping to work the new Brain Initiative devices into our distribution efforts as soon as possible. So I like to end my talks talking about how we're really not the only game in town for neuron-sized electrodes. So I think we were the first to do like an array of linearly spaced, you know, neuron-sized electrodes. But there's definitely other electrodes out there. So the mesh probe from the Liber Lab is pictured on the bottom left. That has, you know, very small elements, and you can see there it's living happily in a field of neurons, with the neurons have normal density. The G-Lab did a 10 micron cross version of this, so it's both very small and flexible. And in the upper right there, you can see that red line living happily in a bed of capillaries, apparently without disturbing anything around it. On the bottom is devices made in the Malash Lab. And which is another example of a high Young's modulus probe similar to the carbon fibers. And they have showed remarkably little bleeding associated with these electrodes, which makes sense because they're, you know, very small in size, and they might fit between the holes in the cells and not necessarily have to do as much damage to make their way into the brain. So you'll see that, you know, the dates on these electrodes are very recent. This has very much been something that's happened over the last 5 years. Most, if not all of these groups have been funded by the Brain Initiative. And so I think that, you know, in the future, we're going to look back on this time as, you know, the time when engineers figured out how to make a wire live in harmony with the nervous system. And so obviously, these are primarily being developed as neuroscience tools. And that's, incredibly important for trying to understand the brain by recording from a lot more of it at the same time. But I think it's safe to imagine that over the next 10 years, at least some of these cellular scale probes are going to start making their way into medical applications. I think they are, ultimately, we've created other problems, and some of the materials challenges are significant. I think that these kinds of probes are ultimately going to be a lot safer for large scale implementation. So with that, I just want to thank my funding sources. Particularly, I want to thank the NSF and NIH for funding both the development and the distribution of these carbon fiber electrodes. Here's a picture of my lab and all of the people that did the work that I referred to from our own group. And all of our wonderful collaborators, including all of our carbon fiber beta testers. So I'd be happy to take your questions via email, and I want to thank the host for inviting me to participate in this event.